Hi, and welcome to this lesson on statistical tests. And now, if you're one of my students, it's been a while since we've done new content. I purposely left this last little bit of maths till later on because I thought we might be back in school at this point, but we're not. So we're going to plow on. We've only got a few more lessons left of the Year 12 syllabus, and today it's statistical tests. So our aim today is to use both the t-test, that's the first one, and the Spearman's rank test, or sometimes it's called the Spearman's rank correlation coefficient test, to assess whether an observed difference is significant. Okay, so a little recap first of all. 3 things I'd like you to do. Could you sketch, just somewhere rough, what a normal distribution looks like? Can you then sketch another normal distribution with the largest standard deviation, but the same mean? And 3, can you write down the formula from memory from standard deviation? Now that third one might be difficult. Don't worry if you can't do it at all. I'm going to give you a clue in a minute, but first of all I want you to think about it. Okay, so pause the video, see if you can do those 3 things. Okay, let's go. So first of all, a normal distribution. What should that look like? Well, it's going to look a bit like this. So I tried to draw this freehand, so it's not the perfectly drawn, but kind of more or less. Now, one with a larger standard deviation would look like that. Okay, so now we can actually overlay those 2. So if I overlay those 2, you can see the dotted line in the middle, the point at which it is centered, is the mean, which is the same for both. But then the red one has a larger spread of data, which is what the standard deviation really measures. So the third one, could you do it? I'd be impressed if you could, to be honest, because I'm not sure if I could do it from memory. And you won't have to in an exam. You'll have to calculate it given the formula. But let's see if you can remember the formula, if you work it out from these things. Okay, so they're not in the right order. You have to kind of unjumble them. Can you do it now? Pause the video. Have a go. Okay, so what it is, that last one, it actually is this. So that means the standard deviation is equal to that big kind of capital M on the side is the sum. So this means this is the sum. Whoops, the sum of the value minus the mean value squared over n minus 1. So that's what we looked at last lesson. So standard deviation, you get given the formula in the exam, but there it is. S is the standard deviation, that symbol, sum of x is the measured value, and x bar is the mean value, and then n minus 1 on the bottom is the total number of values in the sample, and then you minus 1 from that. Okay, so that is the standard deviation. So also, just a quick recap on normal. You'll remember that last time I got you doing this kind of content. We calculated the standard deviation from some height data from my students. And then we said that if we had more and more and more students, then their heights would start to look like this. And we said that this is the normal distribution. And this is the way that data is normally distributed in biology. So here are some examples in biology. We typically have, things like baby birth weight, pizza delivery times. This is kind of biological because it's, leg powered. And, weight of shrimp hatchlings was another example there. And they all tend to be sort of normally distributed, especially the more and more data we add. We can sort of plot them like this. Now, today we're looking at a t-test, first of all. And the syllabus reference of the things that we need, that we're going to be looking at, are here. Okay, so this is from the module 4.3, from the AS stuff. And down on the syllabus here, we're going to look at this pink box stuff here. We're going to look at we're going to work out standard deviation. We've already done that. Look at the spread of data. And we're going to look at the student's t-test to compare means of data values of 2 populations. And then we're going to look at the Spearman's rank correlation coefficient if there's time as well. So what does the t-test look at? Okay, well, to kind of look at it, to kind of think about it, it helps to represent it visually, okay? So here we've got a red and a green line. Imagine these are 2 normal distributions of data. Let's say that this is, I don't know, let's say that this is the height of a plant in field A, and then the height of a plant in field B. Is there a difference between these two fields? We can see that the mean of the green data, if we look here, is slightly to the left, let's say slightly shorter than the mean of the red data here. But there's a lot of overlap. So was this just random, the difference, or is it really significant? Is one field better than the other? You can make a decision now if you think it's different, but it's really difficult to say without some maths behind it. What about now? What about here if we have the two fields again, but this time there's a bigger difference between the means. The mean of the green data would be sort of over here, quite a lot shorter than the mean of the red data. But we still see there's some overlap in the distributions. Some plants kind of in the green field are shorter than some plants in the red field. Now, what about here? Well, here we can see that there's a real difference, and it looks like all the plants growing in this sort of red field are taller than pretty much all the plants that are growing in the green field. There's a really big difference. So at which point is that difference significant? That is what the t-test seeks to work out, okay? And in order for us to kind of use the t-test, we need to know some things about the sample. We need to know the mean of this sample. We need to know the mean of this sample so we can compare them, and we can then sort of work out what is the difference between the two means, sort of this distance here. And we also need to know the spread of the data. So we look at the spread of the green data, this kind of standard deviation here, and we need to know the spread of the red data, the standard deviation here. And if we have those things, the means and the standard deviations, then we can start to do some statistics. Okay, so let's look at the formula for this. So there's one more example down there. So this is data taken from many millions of people in the US, men's heights in the US and women's heights in the US. We can see that the median and also the mean height for men here, given in feet and inches, is 5'9.5" inches, whereas for the median women in the United States, when this data was collected, was 5'4" inches. So the data overlap because a lot of women are taller than men and a lot of men are shorter than women, and there's a lot of variability there, but the means are distinct. But is this significant? Well, it actually kind of depends how much data there is, and if we're talking about data from millions of people, then even quite small differences in the means are certain to be significant if we have enough people where that data came from, okay? Right, now we get on to the formula for this test. Sorry, before we do that, when would you use this test? Okay, so this is the test that we're using down here, the two-sample t-test, this one. So when do we use that? Okay, well, what sort of data? Well, we need to be talking about measurements, okay? The investigation involves taking measurements, for example, the two heights. And then we would use this sort of test when we're looking for differences between measurements from different samples. So that could either be the male population and the female population, a field, field A, the green field and the red field, or any other sort of set of data. Potentially, you're trying to investigate whether there is a statistical difference in the arm length of two different orangutan populations in two different forests. Maybe you're wondering if these could be different species of orangutan. It seems like this one orangutan in the forest in Sumatra, let's say, has a slightly longer arm than the orangutans in a forest in Borneo, let's say, and we could maybe sort of investigate if it's significantly different. Okay, so the t-test can tell you if there is a statistically significant difference between two means, and there are certain things that have to be true before a t-test works. For example, we normally do it when the sample size is actually fairly low. When the sample size is less than 25, I wouldn't really worry too much about this specific point. This point is a bit more important. The data should be normally distributed for the t-test to work. But in biology, most data we can kind of assume is normally distributed. So we can use this test a lot of the time. Here is the formula. Okay, let's take you through what this means. So the mean of the first sample, x1, and the mean of the second sample, x2. just going to draw this kind of over here. So imagine we've got our two normal distributions. I'm going to do them both the same color here. So we've got the mean of the first sample, that's x1, and the mean of the second sample, that's x2, okay? Mean of x1, mean of x2. So this gives us, this top bit basically measures what's the difference between the two means. And then this seeks to try and understand sort of what degree the distributions are overlapping, because this S1 squared is the standard deviation of that first sample. So I'm going to change my pen color down here. So yeah, this S1 here is kind of a measure of the spread here, okay? And then this S2 here is kind of a measure of the spread here, the standard deviation. This is just a representation. So it's kind of seeking how much overlap is there. And then we divide by the number of measurements in the first sample, that's N1 here, and the number of measurements in the second sample, that's M2 here, okay? Again, this formula you won't need to know. It will be given to you in an exam, but you will need to be able to use it. So in a few minutes here, I'm going to set you some practice ones to do. But first, we're going to do a run through, a kind of worked example, and then I'm going to set you some work on this. So let's do the run through. Okay, so here's the worked example. Does the pH of soil affect seed germination of a specific plant species? Okay, so we've got 8 pots, 8 seeds in 8 pots with soil at pH 5.5. That's group 1. And in the second group, we've got another 8 pots with soil at pH 7. So is this going to affect the percentage of seeds that germinate? So 50 seeds planted in each pot, and the number that germinated in each pot was recorded. So when we do a t-test statistical analysis, we have an idea that there is a difference, but we need to come up with something called the null hypothesis. So the null hypothesis is really something that we're seeking to disprove if we do have difference. So the null hypothesis in this case is that there is no statistically significant difference between the two groups. And that's always the case whenever we're doing t-tests. The null hypothesis is always there is no difference between the two groups. So in this case, there's no difference in germination rate between the two different soil pHs. So we're going to do this t-test and we're going to see, okay, well, do we keep that null hypothesis or have we disproved it by statistics and saying there is too much difference for this null hypothesis to be true? And then we can move on to our other hypothesis, which is that there is a significant difference between the germination of seeds. And we do this by comparing our value of t, which we're going to calculate, to a table of values which are pre-prepared by statisticians. And basically, if the number of t, which is a kind of a measure of the difference, if t exceeds this critical value, then we reject the null hypothesis. Okay. Now we're going to go work through this. You can work it out with me. You can write it down if you like, or you can just follow along because I am going to set you a sheet with some practice ones in a few minutes for you to do. So here's the table. Okay, so remember in each pot here we had 50 seeds, so we're sort of looking at how many seeds germinated in each pot. So pH 5.5, you know, this first pot, pot 1, there were 38 out of 50 seeds, and you can see the numbers down here. In the group 2, which is the soil of pH 7, maybe it's looking like a few more seeds germinated here. Remember, each one's out of 50. So the first step is just to work out the means of both groups of data. Okay, now I've done that here. Here we go. And I've done it to 2 decimal places. So I can, because I'm going to use that mean to calculate some stuff later on. Then we have to work out the differences. Okay, so this can be a little bit tedious because we're going to need to work out the standard deviation. Now, sometimes if you've got a graphical calculator, I'm not the best with graphical calculators, you can just plug all these numbers in and calculate the standard deviation just by entering the numbers, and then it will just come for you. But you should be familiar with how to work it out sort of longhand. So this here, reminding you, you've done this already, is the value minus the mean squared. So this would be 38 So this would be 38 minus the mean, which is 39.13. Work that out and then square it like that. Okay, I'm going to rub that out actually because I've got the values. I'm going to fly in here in a second, so let's just get rid of that. And if you want to pause the video and work these out, you can, or here they are. So those are the values there. Those are the values there for the differences squared. And remember when we work out the sum, we have to work out, well, when we work out the standard deviation, first of all, we have to work out the sum of those differences. And then we have to divide it by n minus 1. So that would be 7 in this case. So just again reminding you of the of how to work out standard deviation. We do the sum of the differences divided by n minus 1, and then we square root. So in this case, we worked it out as 38.88 for group 1 was the sum of all the differences divided by 8 minus 1, and then we square root the whole thing to give us a value for the standard deviation for group 1 of 2.36. Doing the same thing for group 2, total of all the differences squared divided by 7, square rooted, and we have a standard deviation of 3.42. Okay, so now we have both the standard deviations of the two groups, and just going back a slide, we also have the means of the two groups. Okay, so we see that this one has a slightly higher mean, but it also had a slightly higher standard deviation. So now let's take it on to actually using that t-test formula. Here's the t-test formula, okay? So it's the mean of the two samples divided by, this one is the standard deviation of sample 1 squared divided by the number of tests, or the number of samples from the first one, plus over here the standard deviation for sample 2 squared divided by the number of tests for sample 2. Okay, so let's put in the numbers. So here we have the mean, 39.1 minus 43.5. So that's going to give us the difference between the two means. Down here, that was the standard deviation, 2.36. We've got to square it. Divided by the number of samples, that was 8 for both tests. That was 8 and 8. And the standard deviation for the second sample was 3.42. So we've got to square it. Now, be careful when you're working this out in your calculator. It's so easy to go wrong. I'd probably work it out sort of fairly step by step. So if I was doing that with my calculator, I'd probably start by expanding the brackets. So let me just show you what I mean. First, I would do 2.36 squared divided by 8. I'd put that in my calculator, 2.36. And then I'd go squared, and then I'd divide it by 8. So I'd know that would be, this whole value there is 0.696, and so on. Okay, but just be careful, because there's a lot of different operators that can get confused. But if you work it all out, it's eventually going to come out to that. Okay, t equals 2.99. Great. Okay, what does that mean? Well, nothing yet. It's a measure of the difference between the two means. It's kind of like, and I'm just showing you this graphically so you can kind of picture what you're actually doing with the maths. It's kind of like you've got these two different data sets, the blue and the red here, and t is sort of a measure of this kind of space. The difference. How much difference is there? How much can you sort of get in this area between the two means before the kind of distribution starts to overlap? So that's how I picture it. That's not really mathematically accurate, but it's a way of picturing it. Sort of a measure of this difference. Now, to tell whether that difference is enough difference for you to reject the null hypothesis, you've got to go to a table of values. So we compare our calculated value of t, that should be t there, sorry. Compare our calculated value of t with the relevant critical value in the stats table of critical values. Okay, so to do this, you need to look at something called the degrees of freedom, okay? So the degrees of freedom, you need to know the degrees of freedom, okay? And this is how, I don't really know how to explain this. I'm not a statistician. The degrees of freedom is like the number of options there are kind of for things to, for values to be different, I guess. How many degrees of freedom are there? So it is the total number of sample sizes minus the number of different data sets. So normally we're using 2 different data sets. So normally we have n1 plus n2 minus 2, so it's 14, because we had 8, remember? So this was 8, and this was also 8, so 8 plus 8, that's 16, minus 2 equals 14. Okay, so we would use this row of the table when we're looking at what our critical value is. And in this case, the critical value is 2.15. Okay, so what does that mean? Well, our value of t is 2.99. So 2.99 is bigger than this critical value. Well, that tells us something. Our value for t exceeds the critical value, so therefore we reject the null hypothesis. Basically what we've done is we've said that, statistically speaking, there's too much difference between the data sets for them to actually just to be a random variation. So we can now move on to our other hypothesis. There is a significant difference. We can conclude that there is a significant difference between the 2 means. So pH does affect the germination rate for this plant in the range of 5.5 to 7. Okay, so that is your worked example for t-test. Right, I'm going to now set you this sheet here. So this I'm putting on to Teams, so you should have this sheet now. Now it's got a worked example there again of both the standard deviation and the t-tests. Just to point out here that variance is what we call the standard deviation squared. So S squared is the standard deviation squared, or it could sometimes be called the variance, just to point that out. So there's a worked example if you want to work through those. But the questions I want you to do are down here. Okay, so question 1, 2, and 3 there, using the math skills that we've just practiced. Just to give you an intro into question 1, the diameter of leaves from 2 areas of a plant were compared as follows. So we're looking at the leaves from the top branches of a plant and the bottom branches of a plant. We take the same sample size from each, so N1, 16, and N2, 16. Now the mean leaf diameter at the top was 94, but at the bottom was 179. So quite a lot bigger there, 179 versus 94. Now here it's been given the variance there, which is S squared, so there's no need to square this again when you're using the t-test formula. Because remember the t-test formula, oh gosh, can I remember this? It is the kind of x1 minus x2 bar all over, oh my gosh, can I remember this? It is the s squared 1 over n1 plus s squared 2 over n2. Is that right? I can't even remember. Did I get it right? Yes, I did. No, I didn't. I missed the square root. Missed the square root. Silly. Ta-da. There we go. Okay, that's right now. So remember, you will be given this formula in exam, but I've just written it there for your ease. So complete this, questions 1, 2, and 3, then submit it on Teams, and I'll put up the mark scheme for this at the start of your next lesson, which will be tomorrow, because I think we'll finish there, and we'll do correlation tomorrow. Okay? Thanks very much. I hope that was helpful, and see you next lesson. Bye-bye.